Hi, and welcome back to the EDGE tutorial series. This video is going to explain the taxonomy classification module in EDGE. After this tutorial, users will know when to turn taxonomy analysis on and how to grasp the different classification tools available within EDGE. As a reminder, we are running two job submissions in parallel. The first uses E. coli FASTQ files in the PublicData testData directory in the EDGE interface. The second uses data from the SRA run SRR 2164314. The EDGE taxonomy module will perform sequence classification and help determine sample composition. It does this by assigning taxonomy labels to DNA sequences. This can be useful for a couple of reasons. First, taxonomy is going to help identify what is inside of complex samples. Second, this module can be applied when analyzing purified cultures to help detect contamination coming from lab reagents or mishandling of samples. By default, taxonomic classification is turned on for both reads and contigs at the EDGE interface. EDGE implements several different databases and algorithms for taxonomy assignments. By default, all of the tools are turned on to take advantage of their strengths and provide users with cross-validated assessments. The tools vary in sensitivity and specificity, as well as their classification. Gotcha, for example, offers both bacterial and viral databases. The result of all of the read-based taxonomy profiling methods are summarized in comparative views like the radar, plot, and heat maps you see here. Brief descriptions of the tools are listed here. More detailed information for each of these tools and how each is applied to reads versus contigs can be located in the third-party tool section of Read the Docs. We can now take a look at what this module looks like in the EDGE demo. For both of our samples, we can scroll down to the taxonomy module and see that it is automatically turned on. If we click and look at the drop-down menu, we can see that all of the classification tools and different databases are selected. We don't need to change anything to get the desired results, so we will keep all of the default settings as is. Click over to the SRA sample and make sure it's also turned on. Indeed, by default, it is. Okay, the taxonomy settings are ready to go. In the next video, we will be taking a look at phylogenetic analysis.